Michaela, hello. How are you all? Can I get a thumbs up for a sunny day? Hello. Okay, lay down. Not you, him. Michaela says hi. Everybody says thumbs up. Good. Christina, no thumbs up. You angry with me? Christina Iorio? Angry with me. Okay, I can take that. Okay, hang on. Out. Okay, class. Today's lesson is basically cell biology. So we're starting this new chapter, chapter 7, which last time I was right when I said what it was about, but I was wrong when I said the number. I said it was number 10, but it's going to be number 7. So number 7 is about how, we've talked about signals going back and forth, growth factors, stuff like that. This is about how cells just touching each other, pulling on each other, cause things to happen. Like in gastrulation, when that mesoderm slides up along the ectoderm in Xenopus, and cell-cell contacts and stuff like that. So if you've had cell biology with me, this is going to be a lot of review, really. Here is my PowerPoint. So, chat. Get myself together here. Okay, so the first slide is kind of title slide, which you don't have to know anything from, obviously, is kind of giving you an idea of what we're leading towards in this chapter. So, you've got your mesenchymal cells moving, they become mesoderm. Some mesoderm cells become blood vessels. So here's 2 blood vessels, and they end up fusing together, and they become this pre-heart structure, which then becomes this. And now you see these twirly things here. This is all about contractions of the cell that change the shape of the organ. So that's morphogenesis, where the genesis of the shape of the cell. So that's what we're talking about here. So before we get into the actual what it is going on, we have to talk about the things that are holding cells together and the things that are making cells move, because that's what's going to be operative here. And I'm going to be skipping around this chapter because we don't have time to do it all, and there's only some things that I think that really relate to what we've done already. But in general, there are 3 things that are really important here in morphogenesis. One is cell attachment, how cells hold on to each other. Another is cell shape, and cell shape can change based on the cytoskeleton, which is totally regulatable by signal transduction. And cell migration, cell movements. We saw mesenchymal cells that were constantly moving until they got into place. And so we'll talk in the next lecture. The first thing we'll talk about in the next lecture will be that epithelial to mesenchymal transition and mesenchymal to epithelial transition. So that's all kind of wrapped up in here with cell crawling and cell attachment. Okay, so how do cells hold each other? We've got a couple different structures, and these structures are always like grouped together, so you get little spot welds between cells. So cadherins are one group of protein that are connecting cells to cells, and cadherins are really interesting. Oops, sorry, went too far. Really interesting in that they're homophilic, which means they only bind to themselves. So a cell with E-cadherin will bind to a cell with E-cadherin. If you have a cell with E-cadherin and L-cadherin or something, they don't bind to each other. And that's kind of important because that's what's going to hold the tissue of same cells together and make it move across a tissue that's a different cell. So like mesendermal cells sliding across ectodermal cells. They're not grabbing those cells, they're crawling along it, but they're grabbing onto each other. So cadherins are keeping same cell types together. Every cell that expresses the same cadherin will bind to each other. These are calcium dependent, and I think they're going to be regulated through calcium. You're going to let go by getting rid of the calcium. That's a guess, though. I haven't read that yet. We haven't gotten that far in the chapter yet, frankly. But that's my guess because they are calcium dependent. 30 different types of cadherins, and so they can be used, differentially in different cells. Let me take a quick look and make sure I'm not forgetting something here from my notes. So attachment. Okay, so these cadherins are found in two different types of structures. And the big difference between the structures is what these things attach to on the inside of the cell. So adherens junctions are when you've got cadherins grabbing cadherins on the outside, and on the inside they're attached to microfilaments. We'll talk about microfilaments more later, but microfilaments are made of actin, and that's what's in the cortex of the cell that's holding up the plasma membrane is these actin microfilaments. And you can see here, another interesting thing here is they are attached to these microfilaments with catenins, alpha catenin and beta catenin. We'll see gamma catenin in a minute. Beta catenin is that same single transduction molecule we talked about earlier that's broken down by GSK3 beta and protected by AKT. So beta catenin is really important for signal transduction. You can see here it's a structural protein as well. It's helping to bind the cadherin to the cytoskeleton. And kind of a theme we'll talk about quite a bit here is when there's like information going in both directions here, from the cell to the exterior connection, from the exterior connection into the cell. When the cadherins are activated from the inside by signal transduction, they're going to grab onto other cadherins really strongly. And the inside of the cell can change their conformation and they'll let go of cells. So that's really important to be able to regulate that. And in the same way, when they grab something outside, they relay to the inside of the cell that they're grabbing something. And that stimulates the cell's signal transduction to respond to it as well. So there's information going back and forth between the extracellular and intracellular side here based on binding of those cadherins and signal transduction events here in the cytosol. Okay, so your adherens junctions then are cadherins, which are attaching to microfilaments. And we also have desmosomes. In desmosomes, the cadherin binding is heterophilic, which means you've got different cadherins binding to each other. So it's not the same isoform in both cells. And another difference here is you're connecting your cadherin to intermediary filaments rather than microfilaments. And they're being attached there by alpha and beta catenins again. Placoglobin is another. So I haven't told you all the structural proteins connecting these, but and don't worry about them. But the beta and alpha catenins are important. Okay, so here is your desmosome, slide 5. A different picture of the desmosome. Here's your cadherins, and they're actually, I've changed the name here because if you look, that's not it. If you look it up, there's specific cadherins here involved. One's called desmoglein, and the other is desmocolin. Don't worry about those names. We're just going to call them cadherins. But you can see it's heterophilic because you've got a red one coming here attaching to a yellow one. So it's not homophilic like the other way. Like the other ones, I mean. And here we've got gamma catenin instead of alpha catenin. Although in the last slide I said there was alpha catenin. I think this is probably gamma as well. Sorry about that. I usually count on Leo Lee to catch me on those little typos. He's a sharp character, that Leo. Gonna be a doctor, some say. Or I say. Morphogenesis, cell adhesion. Okay, so you've got your cadherins, which are involved in adhesion junctions and desmosomes, and here are integrins. Integrins are involved in focal adhesions and hemidesmosomes. And the integrins then bind to extracellular matrix, mostly collagen and fibronectin. And also probably some proteoglycans or glycoproteins or something like that, but mainly collagen and fibronectin. So your integrins then are heterodimer. You've got an alpha subunit and a beta subunit. Binding sites for RGD sequence of fibronectin. Who cares? So on the outside, it's attaching to the cytoskeleton or the extracellular matrix, and intracellular attaching to the cytoskeleton. And moving on to slide 7, just like our adherens junctions and desmosomes, there's signal transduction going on here as well. So you've got attachment to the collagen here, which is then signaling to the inside of the cell that the cell is attached. And you've got signal transduction from the inside that tells the cell to either grab it or let go. So we'll talk a little bit later today about cell crawling. And if you had cell biology with me, we talked about it, where these integrins and focal adhesions grab onto the extracellular matrix and hold, and then the cell pulls itself along using that connection, and then it lets go, and then it moves forward and grabs somewhere else. So that's what so these integrins are really important for cell crawling. And again, they're like all focused into spot welds, and in terms of these integrins are called focal adhesions. And here's a signal transduction molecule, focal adhesion kinase, that's really important when these things bind. If you're in cell biology, we talked about cell survival and how it needs certain trophic factors or survival factors to stay alive. And when the cell is attached to extracellular matrix, that activates the signal transduction to tell the cell to stay alive. If you take these cells out of that context and they're not attached to anything, they go into apoptosis. I don't know if that's true in these developmental cells because they're already pretty embryonic. But if you were talking about an adult cell and you took it out of its milieu, little French there for you, take it out of its tissue, and it was no longer connected to collagen, it would go into apoptosis. Okay. Morphogenesis, force generation. Okay, so again, cell biology. Aren't we glad we had cell biology before this class? I've been talking to Dr. Parker about this. It seems like cell biology is like so much easier than developmental biology, and so much of what we did in cell biology now is important in developmental biology. Developmental is a 300 class, and cell is a 400 level class. I think we got to switch those somehow. Excuse me. But anyway, I'm sorry I keep burping, but here I am in quarantine. All I do is eat. So we've got 3 different types of proteins involved in the cytoskeleton that are going to be important in force generation. Less so your intermediary filaments. They're not really used in force generation. They're going to respond to and absorb some of the forces that go on when things get stretched, but they're not actively generating any force. But you've got your microtubules and your microfilaments, and they will be involved in movement and force generation. Microtubules are stiff tubular structures. They're rapidly polymerized and depolymerized, especially right here in our embryo, right? Because the cells are constantly dividing. So they're making their mitotic spindle and then taking the mitotic spindle apart on a regular basis. So these things are forming and unforming and changing. They're very dynamic. And you get movement with microtubules because you've got these motor maps, which are little molecules that walk along the microtubule. Normally in a cell that's not actively dividing, you've got motor maps moving vesicles around in the cells. taking proteins to where they need to go, taking lipids where they need to go. There's a lot of different movements that happen here with these molecular motors. But also these molecular motors are involved in the cilia, dynein especially, causing the whip-like motion of the cilium that makes it move a fluid. So remember in the last chapter we talked about in the mouse embryo, the little cup and the cilia beating fluid in one direction, making a concentration gradient of nodal. So here are our cilia, and the dynein are affecting that. So that's one thing, our motor maps and our microtubules. So they'll be involved in movements in the cell. If you remember way back to, I know you don't, so I'll just tell you about it. If we go back to chapter 4, we're talking about cortical rotation in the xenopus. That the cortex is microfilament. It's the outer gel-like part, and then but the motion is created by microtubules and molecular motors or motor maps. So that's an example of motor maps being used for movements and development. Okay, microfilaments, thin, rope-like, flexible, made of actin. What's to go wrong? These are wonderful little products, and I think you should go to my website and buy them. What are they used for, though? They're used to hold things in place a lot and connect things. Also, they're used in movement of the whole cell. We see a lot of crawling in development with mesenchymal cells coming in through the node and crawling out and moving places. That's all cell crawling, which involves these overlapping anti-parallel microfilament strands. And here you can see the red strands here are those, and those are called stress fibers. They overlap anti-parallel, and between them, there'll be a molecule of myosin. Myosin will grab them both and then make them move like this, and then the cell crunches up, and then it reaches out and crunches up. So it's kind of pulling the cell forward using that. And we also have microfilaments in these gel-like forms. I've said it about 3 times today already. The gel actin that's in the cortex of the cell, here again lit up in red, on the outside of the cell, holding the plasma membrane in place. And your integrins and cadherins are attaching to that, and that's holding everything together. And they're a mesh. This upper picture here is talking about how the fibers just overlap in 3 dimensions and it becomes like a gel. Okay, and that we find in the lamellipodium. Lamellipodium, if we go back to 13, out here is the big broad foot at the leading edge of the cell as it's crawling. Excuse me. Here's another view of that. So here's the whole cell. It reaches out this lamellipodium. You look at it from up on top. There's a shot of the gel mesh, and in front of this gel mesh, you're polymerizing microfilaments and pushing the membrane out. Behind it, you're depolymerizing and then forcing those monomers forward to keep moving in that direction. So that's reaching the foot out. But then underneath, we have the stress fibers, which are pulling the cell forward. You can see here's the nucleus, the whole thing gets pulled forward, and that's stress fibers grabbing the cytoskeleton and pulling the cell. And these little yellow dots here are the focal adhesions, which get formed and unformed. So here the lamellipodium reaches out and it makes a focal adhesion with its integrins. It grabs the collagen there, and then back here, after it's pulled forward, it lets go. And those integrins can get moved back to the front so you can keep on walking in that direction. So cell crawling, really important in mesenchymal cells. Mesenchymal cells. Okay, what else do we got? Oh, that's it. That's all I came up with for today. I guess the next part, I stopped there because the next part kind of starts a whole nother, like the first part's all kind of setting up what we're going to be talking about, and then the next part is actually talking about it. So I stopped there. How you doing, Nick Mather? Are you angry with me? Nick? Give me a thumbs up, thumbs down. Meh, nothing, Nick. You're not paying attention, are you? There you are. Okay, good. All right, so I hate to tell you this, guys, but I didn't bring anything else. Do you guys have any questions? Nope. When is the exam? The exam is during finals week. You'll have to look that up on Banner. I haven't looked it up yet. Yeah, I'm not really sure, but it will be during the 13th. Nick says. What day of the week is that, Nick? Do you know? Nickster? Wednesday. Okay. So it'll be Wednesday at whatever time it's scheduled. That's when we'll do it. What else can I tell you? I got nothing. I got nothing. Shall we call it a day? All we've got now, yep, we're getting a yep from Madalena and Michelle. So we've got a, what do we got? That's today, Wednesday. So we've got 2 more lectures and then the exam. So that final exam will be the second half of chapter 5 and whatever we cover of chapter 7. I'm just going to pick out a couple of topics that match up with things we've already talked about, just kind of looking at it from a different perspective. So that'll give you a little more, that'll make you all more well-rounded. All right, so I'm going to say goodnight. Say goodnight, Gracie. Good night, Gracie. All right. And